Hi, everybody. Welcome back. In this video, we're going to introduce one last modeling approach, one that is still able to capture for some concept of spatial variation and spatial structure in a biological system without the complexity involved in a full reaction diffusion model. This is so-called compartment models. Let's begin by switching over to the slides where we can discuss this further. So the need to iterate over both the temporal and the spatial dimensions of a reaction diffusion model makes that kind of simulation really very computationally costly. You even saw in the last video when you were watching the development of the evolution of these patterns in an activator repressor pair, that it takes time. Now, those were fairly simple systems, only had two components, and they were relatively small domains. And still, it takes on the order of minutes to get a calculation. When you have multiple dimensions, if you move into 3 dimensions, if you have many, a larger spatial domain that we're working with, it can become very costly to do that kind of simulation. It's also harder to implement them. There's a lot of other issues that can arise. Compartment models allow us to capture some of that same spatial structure, some of the important spatial features in the biological system without the complexity that comes from a fully spatially resolved model where we're really trying to keep track of where everything is in space. The idea behind the compartment model is that we break the system into a small number of individual compartments. In some cases, only 2. In some cases, maybe a dozen or so, but relatively small generally. We're not usually dealing with hundreds of compartment systems, although there's no reason that you can't extend compartment models to that larger system size. And we treat each compartment with mass action kinetics. And then some species are allowed to flow between compartments according to certain rules. Okay? Now, in some ways, a reaction diffusion model is a compartment model. You can sort of think of each of the little boxes as being a compartment, and the rules for flowing between those compartments are the diffusion term. But when we talk about compartment models, we tend to mean systems where there really is a smaller number of compartments, and also where there is some natural association between the meaning of a compartment in the model and the meaning of a sort of compartment type structure in the biological system. Reaction diffusion systems can break up even just a continuous domain without any real structure to it into individual little boxes, and that doesn't really map onto the idea of a compartment in an intuitive sense. One place where compartment models are particularly useful for is in simulating behavior in eukaryotic cells. Eukaryotes, including mammals like ourselves, have DNA confined to the nucleus, which is surrounded by an envelope that blocks unregulated diffusion in and out. Ribosomes are confined to the cytoplasm. And what this means is that in order for transcription factors that are made as proteins on ribosomes in the cytoplasm to have an effect and control transcription, they have to enter or flow into the nucleus. RNA must exit the nucleus after it's been expressed in order to, or after it's been created in order to be translated. So there's a natural mapping of sort of the compartments of the cell onto potential compartments in the model. We can look at nucleus, cytoplasm, we can add in endoplasmic reticulum, and other compartments, depending on the problem that we're trying to look at. So how might we do this? And let's focus on, just to illustrate the principles, focus on a simple problem of just looking at gene expression of a cytosolic protein or of a transcription factor that needs to enter the nucleus, and not worry about the other potential compartments in the system. So we may create two compartments, nucleus and cytoplasm. These two compartments are going to have different sets of equations in them. Each of them we're going to model as a mass action kinetic system, but they'll have different equations within them because they have different components that are allowed to be there. Transcription can only happen in the nucleus. Translation can only happen in the cytoplasm. So that's one of the differences. It's possible that we may have different rates of degradation in the two compartments. It's possible that we'll have different transport rules for how different species can move between the different compartments. Right? And so this leads to a different model in some sense in each compartment. Now, we need to be able to account for flow between compartments. As we just said, in order for gene expression to work in eukaryotes, RNA has to migrate out of the nucleus. Transcription factors have to migrate into the nucleus. We have to have some way of accounting for that. Now, actual transport in biological systems ranges from very simple, passive, diffusion-ish driven processes to highly regulated, energy expending processes that transport material sort of independent of gradients and so on. And so there are many different types of models that we can use to describe flow between compartments. I'm going to introduce just one that's a reasonable model for passive transport in the direction of naturally existing gradients of concentration. And the motivation for how we're going to do this comes again from Fick's law, just as the flow term, the diffusion term in the reaction diffusion model did. But we're going to focus on the first law representation of that. Fick's first law says that the flux or the rate of flow per unit area in a system is directly proportional to the negative of the gradient. And so we can write a flow vector that's in the direction of the gradient, or the negative gradient, and proportional to its magnitude. And in many ways, in the compartment model, flow is exactly what we want. We want to understand how much material is passing, flowing from one region to the next. And so this is a natural place to start. Now, we don't have a continuous gradient or a sort of a gradient in the mathematical sense or the calculus sense of a vector of partial derivatives, because we don't have a continuously changing concentration. We have one compartment that has a concentration and another compartment that has a different concentration, and there's not a continuous variation between them. You can sort of think of there's both a conceptual wall in our model, that there's a barrier between the two, and in the context of a cell, there's actually a physical barrier, the cell membrane, or the nuclear envelope between the two compartments. Right? But what we do have are differences in the concentration between the gradient. Right? And this also is can be thought of as a type of gradient, as a differential. You might, and a potential source of energy, a potential source of flow. Right? You might make a comparison that in a downhill flowing river describes a continuous change, a continuous variation, a continuous gradient in a physical sense. A dam which holds water against a wall, and the height of the water is much, much higher than the water at the river at the base of the dam. This is a kind of more kind of structured, but there's still a potential for flow through that process. It's just the flow is almost instantaneous, goes straight down to the other side. And so this, and the height of the well, the height of the difference of the height of the water in the dam compared to the outflow determines how quickly water will pass through if the floodgates are open. Okay. So we're going to take a similar approach. We're going to be motivated by this Fick's first law, and define the flow rate as being proportional to this differential. That is, proportional to the difference in concentration outside and inside the compartment. And we're going to focus on developing our description of what happens when we're inside a compartment, and looking outside to another, or from compartment one to another. Right? And when we can think of this either in this case, we're going to think of this as a symmetric process, where this can sort of describe flows in both directions. When the concentration outside is greater than the concentration inside, this difference is positive, the flow is positive, and we're going to call a positive flow an out to in flow that increases the concentration inside the compartment. And when the concentration outside is less than the concentration inside, this difference is negative, the flow is negative, movement of material out of the compartment. Now, this describes flow, how much material is passing through from one compartment to another as a function of time. But that's not exactly the same thing as a rate of change for concentrations. And our models have all been built around wanting to describe things in terms of rates of change of concentrations, derivatives of concentration with respect to time. What we also need to do in order to convert a flow into a change in concentration is to account for the volume. So if we have one large compartment and one small compartment, a flow of material from one to the other might have a very small impact on the concentration of the large compartment, but a large impact on the concentration in the smaller compartment, right? Because a fixed amount of material has moved from one place to another, but the volumes are different, and that's an essential part of the concentration. We can make this conversion really quite simply by dividing through by the volumes. So we can just say that the rate of change of the concentration inside is this flux, this flow, divided by the volume inside. And so if the volume is smaller, an equivalent flow leads to a greater change in concentration. A larger volume leads to a smaller change for the same flow. And of course, the flow was given by this proportional to a difference in the concentration. So we have a simple form for the rate of change of the concentration inside this compartment. Flows always connect one compartment to another, and so we can make this kind of in-out association. And that means that there has to be a coupled change in the concentration outside in the other compartment. It has to have the same base flow, sorry, but it's normalized, adjusted by a different volume, by the volume of the outer compartment. Right? So same numerator, but a different volume in the denominator. All right. So we have these descriptions for equations that describe the rate of change of concentration due to flow between compartments. And just as in order to develop the reaction diffusion model, we needed to start off with a way to describe diffusion. And once we had that, we could write diffusion in terms of express the effect of diffusion as a rate of change of concentration. We were able to just add that to the reactions. We can do exactly the same thing here, right? So we can just add these terms into our mass action kinetic models to get a complete answer. Now, let's think a little bit more about these equations before we go and do that. In the simplest sort of transport case, we might have a single flow constant for each species. Right? What this means is that flow is passive. It just goes from regions of high concentrations to regions of lower concentrations. The direction is not biased. That means that flow in one direction is equally likely to as flow in the other direction if given the appropriate concentrations. Different species, however, will certainly have different flow constants. Some species are very membrane permeable and can cross a compartment wall more easily. Others are very, very highly restricted and may even have no flow. Right? However, it is not uncommon for the flow constants from in to out to be different from out to in. There can be transporters that are passive, that is, that they act based on the gradients in the system, but that have a structural bias to them so that they move more easily in one direction than in the other. So sort of ratchet-like behavior, if you will. Or one-way valve kind of behaviors. These are things that can occur. In even more complicated systems, this flow constant may not actually be a constant, but rather might be a function of other things in the system. Right? This is when the passage from one compartment to another is regulated by other components in the system. Right? All of these things are details that can be added into the model, and they add complexity, but they don't change the fundamental structure. Okay, so how do we then put all of this together? Let's start with the cytoplasm. The cytoplasm is where translation can occur. So let me make that little correction of this typo here. Protein concentrations are affected by translation, degradation, and flow. And so we might write a rate of change for the protein concentration in the cytoplasm, which has our all of our sort of key terms. There is a positive term that corresponds to synthesis. There is a negative term that corresponds to degradation. Both of these have the common forms that we've seen. The synthesis of protein depends on the RNA concentration in the cytoplasm. The degradation of cytoplasmic protein depends linearly on the protein concentration in the cytoplasm. And added on here, that's a new piece for a compartment model, as different from a mass action kinetic model, is we have a flow term. Our constant normalized by the volume of the cytoplasm and the difference of the cytoplasm in the concentration in the nucleus and in the cytoplasm. The only thing we have to be a little bit careful about here is making sure that we've ordered this appropriately. And it's easy to check. If the concentration in the nucleus is higher than in the cytoplasm, then this term here will be positive, which means the concentration goes up. That makes sense. If you had more in the nucleus, you would have flow in this direction. If the cytoplasmic concentration is higher, this term is negative. We have flow out of the cytoplasm into the nucleus, decreasing the cytoplasmic concentration. All right. Now, that's for the protein piece. The mRNA piece is going to be a little bit different, because for mRNA, only degradation and flow are things that we need, that we are going to be considering, right? Because there is no DNA in the cytoplasm, we can't have transcription in the cytoplasm. So for the RNA piece, we have our degradation term, we have our flow term, which looks very much like the flow term for the protein, just with a different constant involved. And we do not have any synthesis term because there's no mRNA synthesis in the cytoplasm. And in a multi-gene system, every gene would have a pair of equations that looks very similar to this. What about the nuclear component? The nuclear compartment is going to, in some sense, have the opposite structure. In this case, the protein concentrations are the ones that are affected only by degradation and flow. So for the nuclear rate of change in protein concentration, we have our degradation term, we have our flow term, we have now the nuclear volume in the flow term, and I've switched the order of the cytoplasm and nucleus here, just so that we can be consistent and always use a plus. So when cytoplasmic concentration is higher, this is positive, meaning a positive rate of change in the nuclear concentration, which is what we want. This is simply the negative of the flow term from the cytoplasmic side, but with a different volume. Okay. For mRNA, however, this is now the case where we do have transcription allowed. And so for mRNA concentration, in the nucleus, we will have a synthesis term, a degradation term, and a flow term. Again, important to note that the degradation terms have the nuclear concentration that we're worrying about, and the same thing for the synthesis piece. I've used a generic nu X notation here for mRNA synthesis, because in all the models we've discussed so far, this is where all of our control has been, right? And the exact function for the transcriptional rate will be determined by what kind of regulation do we have. Is it unregulated? Is it cooperative or not? Activator or repressor? A single transcription factor or multiple transcription factors? All of these things will affect this term, but using the same sorts of models that we've already seen, but importantly, using the concentrations of the transcription factors in the nuclear compartment, not those in the cytoplasm. Transcription factors in the cytoplasm don't have a direct impact on transcription. They might have an indirect impact by affecting the flow of material in, but they don't have a direct impact. And that's really about it. We, in much the same way that we add on a diffusion term to mass action kinetics to create a reaction diffusion model, we, in a compartment model, we add on a flow term to connect the compartments. Right? This flow term is essential. It's what connects the different compartments and makes them one system rather than a collection of independent systems. Compartment models are kind of an intermediate system, if you will, that let you capture some of the spatial structure that we know is inherent in biological systems without all of the complexity of a reaction diffusion model. And they are particularly well suited to systems that have inherent compartmentalization. Eukaryotic cells fall into this category. And in my own research, I've done work with compartment models of the Golgi apparatus. But so do clusters of cells within a tissue, various tissues within an organ, the organs in a body, and so on. Compartment models fit within a lot of different biological contexts and can be a very, very nice way to capture some spatial variation, some of the organization structure of biology without having a fully blown, fully spatially resolved, and very complicated model. All right, that wraps up this video. The next video is really just going to summarize what we've seen in this first section of the course, the first 3 modules of the class. Until next time, take care and be well.